Hello, who's here today? Hi, Jawar here. How are you doing? Nice to see you. It's our last lesson today, last summer school lesson. Give me a hey in the chat if you're here so I can see you. I know Jawar here's here, but who else have we got? Just let me know. I'll wait for a little minute to let a few more people to join, hopefully, and then we'll get started. Hey, Zainab. Okay, we've got a few more people. This is good. Hey, Monkey's here. Okay, we've got our last lesson. I'm feeling a little bit sad. I've really enjoyed teaching you guys. You've been great. Um, because we've just done DNA and we've done DNA replication, I thought it would make sense to do mitosis. And it's quite a good topic because you've done it at GCSE, but you're going to do it in a lot more detail at A level. So it gives us an opportunity to just add on to what you already know. So let me know, are you ready to get started? I'll put the first slide on because the first slide is really just recall. So GCSE recall in terms of cell division. At GCSE, you did 2 types of cell division. So I was hoping you'd be able to tell me in the chat the names of those 2 types of cell division and anything you know about the differences between them, what they make, how they work. Good. Okay, let's get started then. So we've got the 2 types of cell division, mitosis and meiosis. Okay, so let's try and add on a little bit more. What do these types of cell division make? What are they used for? Yeah, so mitosis, Zainab's saying mitosis is like replication. Yeah, they both are though. They're both types of cell division, right? So they both involve replicating cells. So see if you can say anything else, Zainab. Jawar here says meiosis is for gametes. So let's get that down. What's a gamete? Mitosis is for growth and repair. Mitosis makes 2 daughter cells. So what about meiosis then, Zainab? So Jawar here, gametes, they're not embryos, but they are eggs and sperm. They're sex cells, aren't they, gametes? So they're not embryos because an embryo would be formed after fertilization. So an embryo would be formed after a sperm cell has fused with an egg cell and then started to divide. Meiosis makes 4 daughter cells. Good. And someone said as well that meiosis makes haploid cells. Yeah, and Kira says mitosis makes diploid cells. Good. Good. Yeah, Akshar, mitosis produces the diploid cells, just to get it the right way around, whereas meiosis makes haploid cells. Can anyone remember what a haploid cell is compared to a diploid cell then? What's the difference? Because we're saying mitosis makes the diploid cells, meiosis makes haploid cells. So what's the difference there? I feel like my voice is going today. I've got a bit of a sore throat coming on. Haploid, yeah, half the chromosomes. Okay, yeah, H for haploid, H for half, yeah. Because it only has single chromosomes. So you end up with half the number of chromosomes. Yeah, whereas diploid cells have a full set of chromosomes, or you could say the chromosomes are in pairs. Yeah. Yeah, good Jawar here. Jawar here's just given us the numbers there for the human cells. So a human body cell has 46 chromosomes, whereas a human sex cell or gamete only has 23 chromosomes. Meiosis does PMAT times 2, like that, Kira. Yes, it does. It does. We're actually going to focus on mitosis today. So we're going to focus on the stages of mitosis, which Kira has said is PMAT. Yeah, that's a good way to remember it. But meiosis does have two divisions, whereas mitosis is just one division. Nice. Yeah, and then Jawahir says, when they combine, it makes 46 chromosomes. Yes, so meiosis, which Jawahir is talking about, makes gametes, and then when the gametes combine at fertilization, it gives the full set of 46 chromosomes, and that goes on to make an embryo. Hey, flying Valua. Right, okay, so this is kind of what we know so far. As I've said, we're going to focus on mitosis today, okay? So remember, mitosis is kind of like just your normal cell division. It just makes normal cells, like body cells. Yeah, just normal cell division. So it's not making gametes, it's just making normal body cells, okay? Now, mitosis is part of what we call the cell cycle. But the cell cycle has 3 stages. So let's just remind ourselves what they're all called, because it's not just mitosis. Okay, so it has 3 stages. Is there a nucleolus in an animal cell? Yes, inside the nucleus there is, yeah. That's fine, don't worry. I think we kind of did that in lesson 1. We focused on the nucleus, if you want to look back at that lesson. Okay, so the cell cycle has 3 stages. Stage 2 is what we're going to focus on today, which is mitosis. Can we remember the names of the other stages? So stage 1, which comes before mitosis, and then stage 3, which comes after mitosis. Can we remember the names of those stages? Interphase, which we mentioned last lesson. We mentioned that last lesson because that's when the DNA replicates during interphase. And then Esther says cytokinesis is stage 3. So we're going to focus on mitosis today, but obviously we'll have to mention interphase and cytokinesis because they come before and after, so they're linked, yeah? But together, these 3 phases form the whole cell cycle. Good, okay. So we have interphase. Now this is stage 1. So this happens before mitosis. Then we have mitosis, which is all of these stages. This is why Kira said PMAT. If we remember PMAT, it's going to help us to remember the stages of mitosis. So what that stands for is prophase, metaphase, anaphase, and telophase. Now, I know Kira has, but who else has actually heard of these stages before? Because these are the stages of mitosis. It's kind of like we're breaking mitosis down further into 4 stages. Havana says, I have. Me, nope. It says person 1. Yeah, you might not have, because at GCSE you don't have to learn about the individual stages, but I suppose it depends on what exam board you've done and it depends on your teacher, right? But what we're going to do now is we're going to focus on mitosis and we're going to look at the individual stages and we're going to write a short description of what happens during each of those phases, okay? So let's try to remember PMAT, prophase, metaphase, anaphase, and telophase. Yeah, exactly, Jawahir. We're going to go through that whole process of actually separating the chromosomes. Why are you saying bye? Where are you going? Okay. Before we look at those stages, let's just remind ourselves of why mitosis is so important. There are basically 3 uses of mitosis. Can anyone give me one of the uses? I think you've already kind of touched on this in the introduction to the lesson. But mitosis is needed for 3 things. So Kira says growth, repair. So repair of damaged tissues. And the other one, the other R, is replacing cells. Obviously, these 2 link together, right? Because we do replace cells to repair damaged tissues, okay? So, yeah, I do sound a bit funny today. I've got a bit of a sore throat. but I didn't actually realize until I started talking, and now I'm talking a lot, I can feel it. Um, so yeah, we're replacing cells to repair damaged tissues, but we're also using it for growth. Yeah, that's how organisms get bigger. That's how we all grew from a baby to a toddler to a child to an adult, cell division through mitosis. Yeah, it's even how we grow from a zygote or a fertilized egg cell to an embryo, then a fetus, then a baby. So growth is obviously one of the most important things for mitosis. But even when you've finished growing, mitosis is still happening, yeah, to replace cells and repair damaged tissues. So these are the three reasons why we need it, if you like. I've got a coffee jar here, I'll be okay. Should we go through the stages then? So we're going to go through PMAT. I've just split it into 2 slides, so we've got enough space to write. Hopefully you've got enough space in your workbooks. So the first stage of mitosis is called prophase. So what we're going to do is annotate on the right-hand side the key things that occur during prophase. And obviously you can see some of them on the diagram as well. But the first thing we're going to write down is the nuclear envelope breaks down. So during prophase, at the beginning of mitosis, the nuclear envelope breaks down. So you can no longer see the defined nucleus because it hasn't got that membrane around it anymore. Okay, so the nuclear envelope breaks down. The second thing we're going to say is, and I hope you're writing this with me, the chromosomes condense and thicken. And this means they become visible. You can see them, okay? Now, this is quite interesting because if a cell is not in mitosis, you can't actually see the chromosomes because they're not condensed, they're not thick enough to see them. Now, during mitosis, they get basically shorter and fatter, they condense, and they become visible. So if we can see the chromosomes under the microscope, we know that the cell must be undergoing mitosis, okay? Because you can't see them otherwise. And we've also got the centrioles move to opposite poles. So these are the 3 key things that happen during mitosis. Oh, is this the garlic onion practice? Yeah, what you can do, and you'll do it in year 12 if you've not done it yet, is you can take the root tips. So if you get garlic and you grow the roots, you can take the tip of the root and you can stain it. So you can stain the chromosomes, usually with a blue stain, and you can actually see the chromosomes, and then you can look at it under the microscope and see how many cells are in mitosis and what stage of mitosis they're in. We don't use iodine for this one. We use a blue dye that specifically stains DNA. And it's so we can actually stain the chromosomes specifically. So we use a specific dye. Okay. You did it in year 9 for a personal development lesson. That's so random. It's like an A-level biology practical. That's so random. Okay, so that's prophase. So we're not separating any chromosomes yet, but we're preparing to do that. Okay, so the chromosomes are getting thicker. They're now visible, and the centrioles move to opposite poles or opposite sides of the cell. This is just one of those topics, you have to just learn it, guys. You have to just remember what happens during each stage because you could be asked to give a description for each stage. Okay, should we move on to metaphase? I always think metaphase is easy to remember because I think M for metaphase, M for middle. That's how I remember it. So during metaphase, and it's really easy to spot this stage under the microscope as well because you can see the chromosomes lining up. So it's the easiest one to spot when you're looking for stages. So the chromosomes line up. We don't say middle though. Does anyone know what we say instead of line up in the middle? We say something else. We call it something else. line up at the center. Not quite, it could be center, but the scientific term is the equator. The equator of the cell. The metaphase plate, whoa. Don't even need to call it that at A level, but you can do. But yeah, it's the metaphase plate is at the equator, yeah? So chromosomes line up at the equator of the cell, and something else we can say for metaphase is they attach to the spindle fibers. Who's heard of the spindle fibers before? Who's heard of spindle fibers? Yeah? Your teacher made you call it that. Yeah, no, you can. You can, Zainab, the metaphase plate, but just bear in mind it is at the equator. Oh, so the spindle fibers, yeah, kind of like ropes. The spindle fibers are actually made of protein, and they're produced using the centrioles. So these are the centrioles here. And we've already said that they have moved to opposite poles, opposite sides of the cell. So the spindle fibers basically extend from the centrioles, and they extend across the cell. And you can see here that the chromosomes, they line up on the equator and they attach themselves to those spindle fibers via the centromere. Do we know what a centromere is? I should have asked that before. Do we know what a centromere is? Have we heard that before? Centromere, no? Okay, I didn't think you would have. So let me just draw a chromosome. It's basically the center. Yeah, it's basically the middle of the chromosome. It's holding the, I'm going to use some A-level language now. I can't avoid it. It's holding the two sister chromatids together. These are called sister chromatids. I need to explain this, don't I? I need to explain it. So before, if you all look at my pen, it's at the top here. Before DNA replication, chromosomes are just single-stranded. Before DNA replication, they just look like this. Yeah, they're just single-stranded. You can't see them. They're just single-stranded. After DNA replication, remember all of this DNA is going to replicate, right? So after DNA replication, the chromosomes are going to look like this. Double-stranded after DNA replication. So after DNA replication, each chromosome has two sister chromatids. Okay, so we've got some new language there. So before DNA replication, we just call them chromosomes, and they just look like single threads. After DNA replication, which happens during interphase, each chromosome now has two sister chromatids, okay, because you've replicated the DNA. So it now has, instead of being like this, it's now like this. And we call each side a sister chromatid. And they are held together by the centromere. Okay, I'm just going to box this off so you can see, yeah? So the centromere is holding the two sister chromatids together at the middle. So it's like the middle of the chromosome. Does that make sense? Can you let me know if that makes sense? Because this is something that students do really struggle with. And it's not really something you've done at GCSE. We know what sister chromatids are. That's in GCSE. I mean, it's not really Jawar here, but maybe we mentioned it at GCSE. I forgot what the word is, but is this when they can like swap part of the chromosome? Yeah, it is. It's when something called crossing over can occur. Zainab says, you think you understand? Yeah. Simplest way to think of it is just with a diagram. So before DNA replication, chromosomes look like this. After DNA replication, remember all of this DNA is going to get replicated like this. Okay, and that's when we say that the chromosome now has two sister chromatids held together by a centromere. Yeah, no worries, Mohammed. Okay, and it's the centromere that attaches to the spindle fiber like this. Okay. Okay, right, so what have we got so far? Let's just recap. Prophase, not a lot is happening. It's just that we can see the chromosomes now because they've thickened, they've condensed. The nuclear envelope breaks down and the centromeres move to the opposite poles of the cell. Okay. Then we go into metaphase. Now at this point, the spindle fibers have formed. So the chromosomes line up at the equator of the cell and they attach themselves to the spindle fibers via their centromere, which is at the middle of the chromosome. Okay. Right, let's see if we can add on a little bit more. And then we'll obviously do some questions at the end to check this. So we've got two more phases left. We've got anaphase and telophase. Remember the order is PMAT. So anaphase is the phase that we all know because anaphase is what we talk about at GCSE. Okay. So during anaphase, I'm going to call them sister chromatids. The sister chromatids are pulled, so they're physically being moved by these spindle fibers, pulled to opposite poles using the spindle fibers. Okay, you can see this happening. This is the easiest phase to see under the microscope because you can see the chromosomes look like V shapes because the centromere has split and the sister chromatids are being pulled by the spindle fibers to opposite poles or opposite ends of the cell. In fact, I'm just going to add on before that, the centromeres split. Okay, so look, let me do you another picture. Here's my chromosome with two sister chromatids. Here's my centromere. When we go into anaphase, the centromere splits and the sister chromatids are going to get pulled by the spindle fibers to opposite poles. Okay, but I'm calling them sister chromatids here because each chromosome consisted of two sister chromatids, and now we've split the centromere, the sister chromatids are pulled to opposite poles. Okay. Right, telophase. This is the final stage of mitosis. Are these the same phases for both plants and animal cells? Yes, good question. Yes, this is the same phases for both plants and animal cells. So it's, you don't have to learn anything different. Okay, so during telophase, the nuclear envelopes reform and two new nuclei form, which you can see here. And at the end of telophase, so I'll put it last so it's in the right order, the chromosomes decondense and will no longer be visible. So this is at the end of telophase. Okay. So they've reached the opposite poles, they decondense, you can't see them anymore, and the nuclear envelopes reform. So at the end of telophase, you've got two new nuclei. Okay, hi. Just joined, don't worry. We're doing quite a hard lesson today. We're doing mitosis. Okay, but that's not the end of cell division. All we've got here is two new nuclei. we still need to separate the rest of the cell. So we still need to separate the cytoplasm and the cell membranes. Okay, so this is where we've got an extra stage after mitosis. So after mitosis, we've got cytokinesis. And this is the third stage of the cell cycle. So at the end of mitosis, we've got 2 new nuclei, but what we still need to do is divide the cytoplasm and form 2 new cell membranes. So let's write this down. The cytoplasm divides and 2 new cell membranes form to create 2 daughter cells, as we call them. Okay, so what are the centrioles? So the centrioles are basically the structures in the cell that form the spindle fibers. Okay, now the centrioles are just in the cytoplasm, but during mitosis they will move to opposite poles of the cell and form those spindle fibers across the cell so that the chromosomes can attach, because obviously the spindle fibers pull the chromosomes apart. Okay, they're basically made out of protein. This is the last biology summer lesson, yeah. But we don't need to know too much about them other than what do we say? We say they move to opposite poles and they form the spindle fibers. Okay, so they're vital for cell division. Okay, so this is cytokinesis. We've now separated the cytoplasm. We've made 2 new cell membranes. We've created 2 daughter cells. Okay, so all together, in terms of what we need to learn, we've got interphase, then we've got mitosis, then we've got cytokinesis. And just so you know what's expected at A level, you should be able to name all these stages in the correct order and describe what is happening during every single one of those phases and recognize them on a diagram or an image taken under the microscope. You should be able to recognize the phases based on what you see happening. So we're going to have a go at that with some questions, okay? Let's just finish off our learning by describing the end products of mitosis. So in terms of end products, we've made 2 daughter cells, and they are diploid, so they have the full set of chromosomes. And what about in terms of variation? Can we remember from GCSE, are we making genetically identical cells here or genetically different cells? Because meiosis is different. Good, so they are genetically identical. Because if you think about what we've actually done here, we replicated all of the DNA. So we had 2 identical copies. One set of that DNA went into one daughter cell and the other set of that DNA went into the other daughter cell. So the daughter cells will be genetically identical and they'll each have a full set of chromosomes. Okay, whereas what you're saying, Suranshu, is meiosis. Meiosis makes haploid cells, 4 non-identical haploid cells. That would be meiosis, not mitosis. Meiosis makes 2 diploid, genetically identical cells. Okay, so that's our end products. And as we've already said, these cells are used for growth, replacing cells, and repairing tissues. Should we have a look at some questions, guys? I was hoping to do a little Kahoot as well, so I hope I'm not going too quickly. I'm just trying to fit it all in. Okay, so these are A-level questions, yeah? These are A-level standard questions, not GCSE, so you might find them a little bit difficult, but we're going to have a go together. Complete the table to show the different stages of mitosis and the order that they occur in. So we need to fill in the names of the stages and we need to number them. So you can see they've already put prophase in there as number 1. They've put metaphase in there. What stage would metaphase be? What number? Prophase is 1, so monkey, yeah, metaphase is 2. Okay, so what would be phase number 3 then? We need the name of the stage for stage number 3. Okay, good. And then obviously we need to fill in number 4. So what would the final stage of mitosis be? Good. Okay, so nice and easy, but this is very similar to what was on a past paper. Okay, so you can get some nice questions like that. Now, if you look here, we've actually got an image taken under a microscope. Okay, obviously the chromosomes have been stained with a specific stain that stains the DNA, so you can see the chromosomes. Which of the cells, A, B, or C, do we think is in metaphase? Let me just letter them. Which one do we think is in metaphase? Okay. It is B. Can you explain why you knew that, please? Can you tell me in the chat, how did you know that B was metaphase? What's the name of the stain? So we used to use a stain called acetic orsin, but I'm not sure if that's used anymore because it does give off quite dangerous fumes. You have to do it in a fume cupboard when you use acetic orsin. So I think there's another stain that is used instead of acetic orsin. I can't remember the name of it. Methyl blue. It could be methyl blue because methyl blue is definitely less hazardous. Yeah, so I'm guessing, Kira, if you use methyl blue, you didn't have to do it in a fume cupboard, no? Okay, so obviously B, because we can see chromosomes lined up at the equator. Okay, yeah, so that's quite an easy one to spot. So which of the cells do we think is in prophase? Now this is kind of process of elimination because during prophase, nothing is really happening with the chromosomes other than the fact that they become visible because they condense and thicken. But process of elimination is going to tell us it must be C because it's not A, right? Chromosomes are visible. They've condensed. Yeah, so we can see them, but they're not really doing anything yet. They've not moved yet. They're not lining up on the equator. They're not being pulled to opposite poles. So it's got to be C. Okay, what's happening in cell A? Now, can you give me the name of the stage as well for A and try and give me a description of what is happening? I will miss you. I'm going to miss you all. You've been really good in this summer school course. Can't wait to do experiments. Yeah, you do. The practicals are quite good in A-level biology. There are 12 required practicals. This is one of them where you have to actually stain the chromosomes and try and view them. Yeah, so I've made this a 3 marker. So I'm guessing one of the marks would be for naming the stage because we haven't actually been told the name of the stage, right? So anaphase, sister chromosomes are pulled to opposite poles by the spindle fibers. Nice. The centromere split. Yeah, let's think about how we could get marks. Centromeres split might get you a mark. Sister chromatids, that's what I'm going to call them because that is more accurate. Sister chromatids are pulled to opposite poles. That could get you a mark. And by spindle fibers would get you a mark as well. Now you can call them chromosomes, okay? They will allow chromosomes are pulled to opposite poles. But strictly speaking, at this point, I would call them sister chromatids because you're going to split the centromere separate the sister chromatids. Sister chromatids are pulled to opposite poles. I would then start to call them chromosomes again once the nuclear envelope has reformed. But you know, they are going to be generous. They'll allow you to just say chromosomes, so don't worry about it too much, okay? Nice. Perfect. So we've easily got 3 marks there. I think there are probably four different things we could say and get those 3 marks. Okay. So what we're going to do here is try and name the stage. What I might do is number them. So you can put the number in the chat and then tell me which stage. Now, it might be a stage of mitosis, or it might be just a stage of the cell cycle, because remember, the cell cycle has interphase mitosis and cytokinesis. Okay, so put the number and then put the name of the stage. CFK, are your A-level lessons suitable for OCR? We have a separate OCR course. Actually, that's a lie. No, in year 12, we have one A-level course that is suitable for both AQA and OCR. The year 12 content is quite similar for AQA and OCR. There are slight differences, but I will highlight those in the lessons. In year 13, the content is quite different, and we have totally separate courses, one for AQA and one for OCR. Okay, but in year 12, it's like 90% of it is the same. So whether you do OCR or AQA, the year 12 course is going to be useful. Okay, let's have a little look. So number 1 is interphase. That's correct. These are all in a random order, so you have to kind of jump about. Number 2, nuclear envelope reforms. That would be telophase. Well done. Number 3, chromosomes line up at the equator, which is the middle of the cell. So that's easy. M for middle, M for metaphase. Good. Number 4, chromosomes condense and become visible. That would be prophase. All that really happens during prophase is you can see the chromosomes, they're condensed, they're visible, and remember, the nuclear envelope breaks down and the centrioles move to opposite poles. Okay, so it's like a little story that you've got to tell for each stage. Centromeres split. Well done, Esther. Anaphase, that would be at the start of anaphase or early anaphase. The centromeres split. Sister chromatids are pulled to opposite poles. Number 6 is anaphase as well. That would be just a bit later on in anaphase, after the centromeres have split. Cytoplasm divides. Good, Angelina. Cytokinesis, which is after mitosis. Nuclear envelope breaks down. Good, Oliver. That would be prophase. Okay. So it's just a case of remembering this. This is mitosis. These are the 4 stages of mitosis. And then we've also got interphase and cytokinesis. Okay. Is this okay for Edexcel? Edexcel A-level. I have less experience with Edexcel. So I would have to say I can't give you a confident answer. In my experience, for biology exam boards, a lot of the content is the same because there are fundamental things in biology. Like, you know, you're all going to learn mitosis. You're all going to learn DNA replication. All the things that I've covered in summer school are going to be on all the exam boards. But if you're talking about the full course, what I would probably do is have a little look online at the Edexcel specification and compare it to the AQA specification. And if you do that, you'll be able to see how much of the content lines up. I mean, I'm guessing it will be a lot. I'm guessing like at least 80% of it will be the same, but I can't give you a guarantee for Edexcel because I'm not as knowledgeable of Edexcel. Okay. Right. Are these phases of mitosis disrupted during cancerous cells, and can it be reversed when so if a cell is cancerous, which basically means it's uncontrollably dividing, yeah, and it's forming a tumor and the cells are spreading around the body, it's basically the cell cycle is happening too quickly. So it's going through the cell cycle more times than it should. So it's dividing too quickly. So it's not like the stages are different. The stages are still happening in the same way and in the same order. It's just going through it too many times. It keeps going through the cell cycle. So it would be going interphase, prophase, metaphase, anaphase, telophase, cytokinesis, and then doing it again, and then doing it again, and then doing it. And it's not being stopped and it's not being slowed down. It's not being regulated. Okay. Right. So if there's the same topic, yeah. So if in EdXL, if you've got a whole topic on, I don't know, biological molecules, which we do on AQA, obviously all of those lessons then would be relevant. You could watch them. But for summer school, all of these lessons will be relevant for EdXL because I've tried to pick topics that are just pretty standard. Just ask me your question in the chat, Saranshu, and I will answer it. Right, let's have a go at this question, maths question. The time required for a cell to complete the cell cycle is 4 hours and 18 minutes. Calculate the time required in minutes for this cell to multiply to produce 8 cells. So I'm going to do you a little diagram that might help you. Might take me a while, bear with me. So we want to go from 1 cell to 8 cells. So 1 division would give us 2 cells, 2 divisions would give us 4 cells, 3 divisions would give us 8 cells. Three divisions needed. And you can check that with maths because 2 to the power of 3 is 8 cells because you're doubling the number of cells 3 times, 3 divisions. 2 to the power of 3 is 8. So that's another way of doing it. The first thing I would do is convert 4 hours and 18 minutes into minutes because I need my answer in minutes. So what's 4 hours and 18 minutes? 60, 120, 180, 240, 258 minutes for 1 division. And I need 3 divisions. Oops. So 250, 500, 750, 758. I'm trying to do it in my head. 766, 774. That's what I'm getting. But I've done it in my head, so correct me if I'm wrong. Saranshu, I hope I'm still saying your name right. At the start of the lesson, I don't know if you were here at the start of the lesson, but right at the beginning, we did a quick comparison of mitosis versus meiosis, kind of GCSE level. So we have been through and talked about, you know, what kind of cells they make, what they're used for, how many daughter cells, whether they're identical or not. I think it was the very first slide that we did. So you can always go back and just watch the start of the lesson again, okay? Yeah, good. That's nice and easy, right? 774 minutes. Just make sure we convert hours to minutes and then multiply it by 3 because 3 divisions would give us 8 cells. Okay. Should we do a Kahoot, guys? Should we have a little bit of fun for our last lesson? I've made a Kahoot. How many marks would this be? 1 or 2, I would say, Oliver. 2 if they're being generous. It might just be 1 because it's not particularly difficult. We're going to give it a go. I've made a Kahoot. It's kind of a mixture of different topics from summer school. Let me see if I can get this going. Let's hope this is the right one. I've made a GCSE one and a year 12 one this morning. I think this is the year 12 one. Let me see if I can copy this link as well. So yeah, it's a mixture of questions. I tried to pick kind of topics that we'd covered in summer school. Not made it too hard, but it is kind of some A-level content in here. There's probably some random questions as well. I think I threw in a few random questions. Okay. I'll just give you another minute and see if there's anyone else that can join. So you can obviously join with the game pin, or I put the link in the chat. I can't actually see how many people are in the lesson, guys. Can someone tell me? So I don't have YouTube open at the minute. Tell me how many people are in the lesson, and then I'll know if we need to wait for anyone else. 30, okay. Wait a little bit longer then. It might be that not everybody can join, I suppose. Even if you can't join the actual Kahoot, obviously you can still stick around, just write your answers down, or even you could even put your answers in the chat so I can see them. But then you might help other people. You need to create an account. I'm not sure how long that takes, Monkey. You might not have time. Oh, Kira says you don't need to. Okay. Just give 20 more seconds and then we're going to start. It says it's a private Kahoot. Have you tried clicking the link in the chat? What you can always do, Monkey, is just put your answer in the chat and you can add up how many you've got right. Okay. I think we're going to have to make a start so we've got time to do it though. Okay, let's go. Okay, it's the right quiz. That's all right. I've given you a little bit longer for this one. I've given you 30 seconds. I thought this was the last question that I'd done. Does it just give them you in a random order or something? Okay, I'm guessing if you got that wrong, it was just because you rushed your answer. I'm pretty sure we all know the base pair rule, right? Nice, easy one. Nice. I should have joined in, did it myself. That might not have been very fair. Seeing as I made it, I think I would have won. Yeah, so we've got phospholipid bilayer, and then remember we have like intrinsic proteins, extrinsic proteins, channel proteins, carrier proteins. It's the cell membrane, isn't it? Okay, question on today's lesson. Yay. Yeah, nuclear envelope breaks down during prophase. Anaphase, that's when the sister chromatids separate. They're pulled to opposite poles. Zainab. Angelina has a streak with 4 correct answers in a row. Well done, Angelina. Did you get in the end, Monkey? Yeah, hydrogen bonds. We have 2 hydrogen bonds between A and T, and then 3 hydrogen bonds between G and C. Just so you know, peptide bonds are found in proteins between amino acids. And phosphodiester bonds join the nucleotides together. So they're between the phosphate groups and the sugars. Aisha is the highest climber. Yeah, Golgi body modifies proteins and then packages proteins into vesicles. Ribosomes synthesize proteins. Roughy R synthesizes proteins because it has ribosomes on its surface. Then it transports them and then packages them into vesicles. Zainab's still winning. Oh, this is quite a tough one. Oh, yeah. I knew that was quite a tough one. Yeah, so a phospholipid, which is a type of lipid, a type of fat that makes up the cell membrane, has glycerol with a phosphate group attached to it and then 2 fatty acids. So we call it the head and the tails, but the head is glycerol and phosphate and then 2 fatty acid tails. Oh, Kira's taken over. Yeah, I was trying to trick you a little bit with this one. I was trying to think of some answers that might confuse you. Yeah, so someone fell for it. I put RNA polymerase in there. RNA polymerase joins RNA nucleotides together, but obviously this is DNA replication. So we're looking for DNA polymerase. It makes phosphodiester bonds. Those of you that put DNA helicase breaks the hydrogen bonds and separates the 2 strands. Okay, that's the other enzyme. Again, I was trying to think of answers to confuse you a little bit. Yeah, so HIV, obviously virus, has attachment proteins on its surface, and the attachment proteins bind to receptors on the cell surface membrane. They don't actually pierce the cell membrane. We don't call them antigens on the virus, we call them attachment proteins. And then I thought reverse transcriptase might confuse you because HIV virus has reverse transcriptase, right? But that enzyme is used to convert RNA into DNA, not to actually help it enter the cell. Angelina, it's good because the top position keeps changing. Right, I think we've only got 2 questions left. Nice one. Yeah. Chromatids are separated or pulled to opposite poles in anaphase. Those of you that put metaphase, metaphase is when they line up on the equator and attach to the spindle fibers via their centromeres. Angelina's going to win. Okay, let's do the last one. Oh, quite a hard one. See who got this one right. Yeah, well remembered. It was a few lessons ago that we did this, but it's T helper cells, which are a type of white blood cell, a type of lymphocyte. All the other answers there are other types of white blood cell. They all do exist, but HIV specifically infects T helper cells because that's the cell that has the receptors complementary to the attachment proteins. Well done. Let's see who's won. So Havana is in third place. Congratulations. Second place, Kira. Well done. Good work. We know who it is, don't we? Angelina. Well done. Good work. Some of those questions were from quite a few lessons ago, so you really had to think back. Well done, guys. Good work. I could have included a few more questions. I was worried about the time. Right, let me close this down. Let's leave that. Well done. Okay. Right, we've reached the end. We've reached the end of our lessons. I'm genuinely quite sad. You were doing so well at the start. Zainab was in the lead. Yeah, we've reached the end of our lessons. Thank you so much for joining me over this summer school. I hope you found it useful. I hope you found it interesting. I hope it's helped you kind of feel prepared for A-level biology. Just to finish with, I think I've been through this with you before, but I was just going to ask if anyone had any questions regarding the full course. Has anyone got any questions? Let's just a reminder that there is a special offer. So that would be the price in orange, 33% off for you guys because you've been in the summer school. The link is in the description to the video, which you know. You can click the link and get chatting with the code SUMMER2024 to get the money off. Money back guarantee if you're still not sure, although you've all trialed the summer school, so you kind of know what to expect. You can think about all the other subjects that we offer. Ultimate Pass, remember, is where you get access to all of these subjects for the full year for a ridiculously low price if you think about how much it costs per hour. We can split it into installments. So yeah, if you haven't done it yet, have a think about it if you think it would be useful to support you. I mean, A-levels are challenging, right? I'm not going to lie. It is a big jump from GCSE biology to A-level biology. So don't leave it too late. If you really want to, guarantee these high marks, it is better to start the extra work and the extra revision at the start of the course rather than leaving it till the end of year 12 or the end of year 13. That's the timetable which I can leave on the board. So Oliver, if you're doing biology, it's Monday and Wednesday. It looks like it's 6:50 to, hang on. Yeah, 6:50 to 7:50, that's right. Could you do the full course instead of go to school? In theory, yes. Yeah, we cover the whole curriculum. Like we do everything. We don't go too quickly. We cover every single topic from start to finish, just like you would in school. We go through lots of exam questions. We even go through all the required practicals and the kind of questions that could come up on the practicals. We go through exam technique and stuff like that. So yeah, we cover everything. Are all the free lessons over for year 12? Yes, I've got a couple. I've got one GCSE lesson left. I've got a couple of year 13 lessons left. But for year 12, this is the end. What will the first lesson be in September? Oh, good question. It's going to be biological molecules, which is one of my faves. We start off with a bit of biochemistry, which I haven't really done in summer school. So we start off with carbohydrates, structure of glucose, monosaccharides, disaccharides, polysaccharides. Then we move on to lipids. So we do all the foundations first. So we do all the different biological molecules that make up living things, so you can then understand the processes when we get onto them. You're going to talk to your parents about it. Yeah. And obviously you Kira, if you're not sure on the exam board, you could try and find out from your school or college, or get back in touch with us in September. What's my personal favourite topic? That's a good question. So anything to do with enzymes or anything to do with DNA and genetics. I love in year 13, there's a big inheritance topic where we do like mad Punnett squares, like mad genetic crosses. I love that topic. That's probably my favourite. But I also love all the detail of like transcription, translation, anything to do with DNA, mutations, that kind of stuff. Yeah. I mean, what you could do, Kira, is just click the link in the description, get into the WhatsApp chat, and just start a conversation and kind of just say, I'm not sure about the exam boards, and see what they say, see if they can like give you the discount, even if you have to wait until September. What if I can't join the main course? I mean, as well as the main courses, we're going to do Easter revision courses, we're going to do cram courses. So you just need to keep an eye out on our social media, and make sure that you join any of the extra courses that we do. And keep following us on social media, because we do a lot of videos to help with GCSE and A level. Can I join with the discount after school starts so I can talk to my teachers? Kira, I can't guarantee it because I know that they're only selling like a limited number of places at the discounted price. However, I think they would try and help you out. They would try and give you priority because you've been in the summer school. So I would just explain, like, I have been in the summer school, I've attended all the lessons, I want to use the summer school discount. I just need to wait until I find out exactly what exam boards I'm doing. And then hopefully that will be fine. Will you be covering the topics that we learn in more detail? Yeah, all the stuff we've done in summer school, we will do again. And we probably will go into a little bit more detail, and we'll do more exam questions. You'll get more homework questions as well with the main course. Okay, right. I'm going to have to say goodbye. Thank you for being such amazing students. Thank you for making it so fun and so easy for me. And yeah, keep in touch. If you're not joining the main course, you can still keep in touch via social media. Let me know how you're getting on. You can always put in requests for little videos if there's a topic that you don't understand, and I can try and film a little video for you. Let me know, yeah, let me know how your remark goes, Kira, definitely. Okay, good luck to you all. I'll be interested to know how you find A level. So yeah, do keep in touch. Thank you guys. See you soon. Bye.